So this section talks about shaping evolutionary theories. So there's different mechanisms for evolution and how it happens. There is this principle called the Hardy-Weinberg principle, and we're not going to go into the math too much on this, but basically this principle states that whatever the allele frequency is, meaning the genes in one population, if basically no evolution or no if the genes remain constant in the population, then in the next generation you should have about the same proportions of each, in this example, type of animal. So if in the first generation here I have a constant gene frequency, and say I have one red owl and two gray owls, then if Hardy-Weinberg is in equilibrium, meaning the gene frequencies are constant and that stays the same, the genes stay the same, then in the next generation I'll have, in this example, double. I'll have maybe two reds, 1 to 2, and then here were two original adults in the first one, so I'll have four of those, so everything just doubled. So that's if everything is in perfect equilibrium. Now that basically almost never happens. So why is Hardy-Weinberg important? Hardy-Weinberg is actually important to look at so that you know what is changing in a population that caused a change in the gene pool. So there's different things that can change that. For Hardy-Weinberg to work, you have to have a very large population. You also have to have no immigration or emigration, no things moving in and no things moving out. You have to have totally random mating where there is no choice in your mate. Then you also have to have mutations not occurring, and you also have to have no natural selection or factors that are causing some to live and some to die. Now, obviously, this basically never really happens. So it's important to look at like what causes where this went wrong to see where changes occurred. So one of the ways that populations can change is because of genetic drift. This is where the genes drift or move to one extreme or to another extreme, or basically the alleles change, the genetics change. So in genetic drift, the diversity of a gene pool decreases due to a decrease in the number of organisms. So there's less organisms, so the genes drift one way or another. Gene flow is when the population increases or decreases, so those genes become more or less prevalent in the group. For the founder effect, I actually really like this one. It's when a small population settles in an area and then builds the entire population from there. So one example of the founder effect is this green iguana. These green iguanas in Hurricane Lenny in the late 1990s, I think 98, 15 of these green iguanas in the hurricane got blown out to sea on a giant raft of trees. They survived eating what they could until they floated 200 miles to another island that did not have these iguanas. These 15 crawled off of the island and started populating the whole thing. And within a year, they saw pregnant females and babies and things like that, that this population had taken off. That is the founder effect. There's going to be very little gene diversity because there was only 15 individuals that started that population. So there's very little genetic diversity, but they founded a new population. Another is a bottleneck. A bottleneck is when a number of organisms die off, and so there's only a few of them left, and then they're left to breed for the rest of the individuals. I just got up to go see the northern elephant seals in Northern California. It was so awesome. But the northern elephant seals were bludgeoned and killed to near extinction in the late 1800s until the Smithsonian. And we've talked about how museums and zoos, their behaviors of the past don't match their newer behaviors of the future, that they do a much better job now than they used to. But the Smithsonian found what they thought was the remaining 8 of these animals on an island in Mexico. They actually bludgeoned 7 of them to death and collected their bodies to go put into the museum in the Smithsonian. One got away. And they thought these were extinct. They were declared extinct. Well, thankfully, a population of about 20 of them survived and remained hidden on a nearby island. But now those 20 that had a genetic bottleneck, meaning there was a lot of them, but then they went through this really narrow gene like pool population, they have ended up breeding to where now they're not even on the endangered species list, and there's over 100,000 of these living all the way from, down in Baja all the way through Northern California. So these are the northern elephant seals. You can also have, so that's called the bottleneck effect. You can also have gene flow. This is where, gene flow is basically genes flowing through a population, and we already talked about that. Non-random mating is another thing that can affect Hardy-Weinberg, and that is where organisms choose a mate instead of just accepting whatever mate by chance is chosen for them. So in northern elephant seals, for example, the one big male will go and fight all the other males, and then he'll develop a harem, and he'll have 10 to 15 females that he guards, and he gets the mating rights and the other males do not. That's a form of non-random mating that affects Hardy-Weinberg. Another thing is mutations, and you can see how mutations coming in will affect Hardy-Weinberg. Another thing is the types of natural selection. So sometimes you'll have selection. This line right here, this dotted line, shows the old population. Sometimes you'll have stabilizing selection, where basically the most common type just keeps going, and the extremes on either side end up dying off. Another thing that you can have is directional selection, where if this was the original population here, eventually it starts to go towards one side or the other, like the ones that are taller, or maybe the ones that are shorter or darker or lighter, but it'll kind of shift to one side or the other. Disruptive selection is when you have something, but it's the extremes that end up surviving. So say you have an insect that can hide on red or white or pink flowers, but all the pink flowers die off. Well, now it's going to be the red and the white that end up surviving, but it's all the pink that dies off. So that's called disruptive selection. So those are all different types of selection. Another thing that you can have is isolation. In isolation, you can have pre-zygotic isolation and post-zygotic isolation. In pre-zygotic isolation, that is something that stops the mating from happening in the first place. This can be a break, like in geography, where the two organisms just aren't even near each other. You can have it where two birds that are very similar in other ways but have different mating songs don't understand each other's songs. You could have instances where one organism is a lot larger than the other or smaller, and so they're not able to mate. In post-zygotic isolation, this is where two organisms can breed, but then the baby is unable to breed. So this would be like here, a lion and a tiger. So that's, makes a ligar or a tigon. And basically with that, their baby can't breed, like a horse and a donkey makes a mule. The mule is sterile. That's called a postzygotic isolation, where a baby can be made, but they're unable to reproduce again. Another type of speciation is allopatric speciation. Allopatric speciation is when there is a physical barrier, like this Grand Canyon here, that's separating types of squirrels. So eventually, these two squirrels become to where a point where they can no longer even reproduce or breed. Another type of speciation that makes new species is sympatric speciation. In sympatric speciation, the two types will live near each other, but say in fruit flies, one starts eating a certain type of fruit, another group starts eating a different kind of fruit. They're both in the same backyard eating rotten fruit that's falling to the ground. Eventually, those two can get to where they can no longer breed. Another type of, like a pattern of evolution I want you to notice is adaptive radiation. Adaptive radiation is when one species gives rise to many other species. Please look at this example in your book. You have a species of fish where they've given rise to 300 different species all in the same lake, and they're all have different adaptations that came from a common ancestor. So another type of pattern that we see in evolution is that you'll have coevolution. Coevolution is when two things are joined together in the way that they're changing or adapting. For example, you can have like bacteria in your own gut. You can have changes in what bacteria is there based on what you eat. All the different bacterias will aid your digestion differently. And then those bacteria now, they're seeing a lot of evidence that those bacteria actually send messages up to your brain on what you crave and want to eat. So if you eat a lot of, junk food, you're going to have more cravings for that junk food because that bacteria, when it, needs it, will send that message up to your brain saying, hey, we need more junk food. And the same thing for healthy food. Another pattern that we see is convergent evolution. And convergent evolution is when several things adapted the same types of grew similarly but in different parts of the world. So solved the same problems but in different ways. And then another pattern that we see in evolution is the rate of speciation. Some animals or plants evolve really quickly due to a change that came, and some go gradually over time. And we see both, but more commonly, it's being accepted that a lot of evolution happens in this punctuated module model where the environment changes, something changes. Something about the environment changes. There's a disease, there's a volcanic eruption, there's, some kind of change that happens, and only some survive, and they're the ones that were fit enough to survive in that situation, and their traits get passed on. All right, so there was so much in there shaping evolutionary theory. Good luck as you progress through that.